Now what we want to talk about is the flowering of the plant. And in flowering, we call it ABC model of plant flowering. So what is the ABC model of plant flowering? Let's see that. This is the wild type flower. Any flowers are just taken as an example. And we know in the flower what we have, it is the ABC model is proposed that there are 3 classes of these organ identity genes functioning overlapping domains to specify. So the thing is, there are different organ identity genes. How exactly the flowering is done? For example, in a flower, if you look at the structure of a flower, the flower structure is composed of carpel, stamen, petals, sepals. Okay, complete flower if you look. And for all these different sections, they are different in their structure, color, nature. They are different. So they are structurally different. They are structurally different because they are coded by different genes, a different way. But there are these different floral identity genes, and they perform to produce different kind of proteins which will interact to provide us carpel, stamen, petal, sepal, and different sections of a flower. But they behave in a combination mode. When one of them is present, it leads to one kind of identity. In combination 2, different identity. In combination 3, different identity. So that's why we need to understand how exactly it's done. See, when the flower is forming, there are different worlds created. 4 different worlds. Okay. In world 1, it's making sepal. In world 2, it's making petal. In world 3, it's making stamen. In world 4, it's making carpel. So you write it as a SPSC. From now on, when we discuss about it, we'll just talk about this sepal, petal, stamen, carpel, SPSC. Now, if you look at here, world 1 is sepal, as I told you earlier. World 2 is petal, SP. And world 3 is stamen, S again. And world 4 is carpel, C. So SPSC. So now imagine one thing, that there are different worlds. Like this is, as I told you earlier, that there are 3 different classes of this organ identity gene we are talking about. So there are these 3 separate sections. This is 1, this is 2, and this is 3. Okay. And among these 3, what we have different combination of floral identity gene, that is A, gene B, gene C. Okay. Now, how they are interacting, based on that we have different. So either what we have this gene A only. So this section is, the number 1 means what? A only, and A only gives sepal. Second one, we have B and A. So A plus B both is giving us petal. The third one, we get B and C. It's giving us stamen. And the fourth one, C only, giving us carpel. So again, we can write it SPSC. S, A only, P, A plus B only, A plus B, and S, B plus C, C, C only. Right? Very clear? This is how 3 different identity genes are. A class genes are known as apetala 1 and apetala 2, known as AP1 and AP2. Both belong to class A genes. Act to specify sepal and petal development, right? So only A sepal, A plus B petal. So presence of A, apetala 1 and apetala 2 determines whether that whorl is going to be sepal or petal. Then class B gene, apetala 3 and pistillata. AP3 and pistillata PI, responsible for flower identity of petal and sepal, stamen, petal and stamen, right? PNS. And so petal and stamen development. The third one, agmos, AG, short form is AG. Acts to specify stamen and carpel development. Stamen and carpel development. So we have this 3 different classes of genes, and these 3 different classes are involved, and in combination they have different floral identity. A little late, so I'll explain this part. Is important because where we are discussing about the 3 different classes of genes, which actually in combination denotes what kind of structure of flower that they are going to produce. We are talking about flowering in Arabidopsis thaliana. And what we want to discuss here is that normally there are these three different floral identity genes. Let me erase one of this. So we're talking about three different classes of genes, and class A, class B, and class C. Okay, that's why we call it as a ABC model of flowering. Okay, so class A gene, full form, there are two types of class A gene. There is known as apetala 1 and apetala 2, AP1 and AP2, part of class A gene. Then class B genes, apetala 3 known as AP3 and pistillata, PI1. And then class C gene is only agamous or AG. There are these three genes, okay? Now understand one thing, that if there are four different worlds during development, there are four different worlds who has different chances or identities depending upon the type of gene expression that they will receive. If they receive gene expression from only A, then it will form sepal. Sepal, petal, carpel, stamen, you know that. So if it receives only identity from gene A, it will form sepal. Then if it receives identity from A and B, both, second word, okay, A plus B both, then it will form petal. If it receives identity from B and C both, third word, B and C both, then it will form stamen. And if it receives only C identity or expression from C gene only, then it will form carpel. This is how sepal, petal, stamen, carpel, all the different four identities are formed. Okay, and you can see this is how the worlds are created. This is A only. Okay, this is A plus B, this is B plus C, and this is C on. Four different sections are denoted. And here you can see A only means sepal, no problem, no gene interactions, only one gene product. And here in the second one, where you have A and B both, so AP1, AP2, both are working. Apetala 1, apetala 2 from A class, and apetala 3 and PI from B class, both are working. Here, what is working? B class means what? That is apetala 3 and pistillata. Along with that, agamous is working, stamen, and C only, agamous is working, carpel. Clear? So, if you know these three different genes and the respective products and how they interact to form four different roles, that will be it. That's all you need to understand to answer the question from this part. So again, different combinations of A and B and C class of genes result in different. So world 1, A alone, sepals. World 2, A plus B, petals. World 3, B plus C, stamen. And world 4, C alone, carpel. Sepal, petal, stamen, carpel, SPSC. Again, remember that way. Now, another important thing from CSIR NET perspective is the mutations in different genes providing different outcome. It's very important to understand the mutations here. So what do you mean by mutation? Here, say this is a wild type. This is wild type. In the wild type, what we can clearly see that there are four different worlds we are looking at. World 1, 2, 3, 4. Genes, how they are interacting. A only, A plus B, B plus C, C on. Right? A plus B, this one is A on. Based on that, we have sepal, petal, stamen, carpel, normal situation, no issues. Now, let's imagine there is a mutation in the class A gene, AP2. Apetala 1, apetala 2, two different types are there in class A. So, if there is AP2 mutation, results in C activity, spreading into worlds 1 and 2, and consequently, homeotic transformations in organ identity with carpel replacing sepals in world 1 and stamen replacing petals in world 2. And why that thing happens? Let's look at here. Where we have mutation in gene A, and if there is mutation in gene A, it's not working. A is not working. Apetala 2 mutation. A is no longer working. So what we have? We have B on the top, C on the bottom now. So what we have? We have C only. We have B plus C, C only. And we know C only means what? Carpel, B plus C, stamen, B plus C, stamen, C only, carpel. So instead of having four different worlds, we are only ending up in two worlds twice. We have two carpels, two stamens. So that is a result of mutation to apatella 2, AP2 in the A class gene. Let's move to the next type, mutations in different genes. Again, the second type. Again, wild type is clear. Now, mutation in the C gene, agamos. Okay, results in class A activity in all four worlds. C genes mutated, so we have only B and A now. There's no C gene activated. So what we have? We have A only. We have A plus B. We have again A plus B. We have A only. So A only means what? Sepal. A plus B what? Petal. A plus B petal. A only sepal. So again, as a result of agamos mutation or class C gene mutation in flowering, we end up in forming two different types of whorls, two sepals, two petals, right? Stamen carpel missing. The third type, we are looking at class B gene mutation, results in flowers with sepals in outer two whorls and carpels in inner two whorls. How? Again, let's look at the same idea here. This is the condition again. So in B, that means there is no B, so only A and C. So A only, C only, C only. A only means sepal, C only means carpel. So what it results? Sepals in outer two whorls, sepal only, and carpel in inner two whorls. And lastly, we have ABC triple mutants produce indeterminate flower only in the leaf-like organs. So there is no flower formation at all. If neither of the genes are active, then there is no conversion of sepal, petal, carpel, all the structures will not form. It'll just stay like a leaf-like structure. Coloring and all these things will be missing. Okay. So this is how the flowering is regulated. ABC model with A, B, C class of genes and the mutation of ABC class and how it leads to the changes. Okay. Now one more thing I must say, that sepalata gene works with ABC genes to specify organ identity. So ABC gene, we know what ABC gene is responsible for. But along with that, there is another type of gene, sepalata. Significant revision of the ABC model was the addition of the class E or sepalata known as ACP. ACP genes, which were first identified in tomato and petunia. The thing is, this is also known as class E, just like class A, B, C genes. This one, sepalata, known as SEP or as class E gene products. Arabidopsis has three SEP genes, SEP1, SEP2 and SEP3 that are functionally redundant. And here you can see how the whole process is working based on the presence of SEP or class E genes. SEP1 and SEP2 are expressed in all four whorls of the flower. SEP1 and 2 expressed in all four flowers, while SEP3 mRNA is detected in whorl 2, 3 and 4. So that actually, now what is the importance of sepalata here? SEP, what is the importance? We know that what in the presence of class A, B, C genes in a plant, there will be formation of carpel, stamen, okay, petal, sepal, all this. But it's important how they are positioned, right? Where each and different organ should be placed, right? So that positioning is kind of denoted by the presence of sepalata and their expression. So depending upon the position, it makes sure that the organization of individual whorls are proper, right? Because obviously there are structures of flowers that need to be embedded inside and structures that are showcasing outside. Other way around, there is no possibility, right? So that is regulated here by this. So SEP1 and SEP2 are expressed in all four whorls, and SEP3, remember, detected in whorl 2, 3 and 4, not in whorl number 1. Okay. In this particular picture, you can see the sepal is present, then petal, then stamen, then carpus. This is how they are organized. And this organization is very, very important in this particular way. And the presence of SEP actually denotes how they will be organized. If stamen is present outermost, that's not safe for the plant because stamen need to be protected. Otherwise, it will be destroyed due to many other reasons. So that's why you need to place it inside. One final thing is you need to know a little bit about the MADS domain protein complexes. In plants, matchbox genes are involved in controlling all major aspects of development, including male and female gametophyte development, embryonic and seed development, as well as root, flower, fruit development. So all the development purpose, whether it's the flower development, root, shoot development, everything are denoted by this matchbox, match domain protein. Some matchbox gene of flowering and plants have homeotic functions like the Hox genes in animals. In animals, we have Hox genes which are homeotic in nature. Now, what is homeotic and what all, we'll discuss about then animal development. In Drosophila development, we'll talk about all these different genes. But similarly, similar to that of the Hox gene, analog are these matchbox protein that are involved in the process of plant growth processes. So this domain family of dimeric transcription factor, okay, all these A, B, C and E genes except for AP2 codes for the match domain protein. Except for AP2, all the other class of A, B, C as well as E, that is sepalata, they are coding for match domain transcription factor. It's an acronym for the first 4 identified members of the family, and they're identified as NCM1 in yeast, AG in arabidopsis deficiens, and SRF in. So based on that, MADS, MAD and S. This is S. You don't need to remember that. But the thing is, these proteins are 56 amino acids DNA binding domain. They're known as MADS domain and bind to CARG box or CA6GG sequences. How it is there? Structure of MAD transcription factor in plants. There are 2 classes of MADS domain protein based on the sequence similarity of the MADS domain. Here you can see MADS DNA binding. So MADS transcription factor, it looks something like that, contains I, K and C 3 different sections. MADS is a DNA binding region, I is intervening region, K is protein-protein interaction region and C is carboxy terminal domain. Binding DNA through conserved DNA sequences called CARG box or CARG box or CCA, CC8086 such and GG. This is the conserved sequence that is present there, okay? So bind this conserved region of the DNA. So they are predetermined for its functions, because if you look at this CC and then 80 such 6 and then GG, it's very long, it's lengthy, still 6, 8, 10 length of nucleotide, and that is a fixed sequence where they bind. So they targetedly bind these sequences and elicit response for specific gene transcription. So intervening regions contributes to the dimerization specificity, and K is the protein-protein interactions form a coil-coil structure also involving the dimerization of the protein, and C, carboxy terminal, act as a transcriptional activation domain in some family members. These are the function of different regions. MADS for binding to the DNA and K is for protein-protein interaction forming coil-coil structure. C acts as a transcriptional activation domain. Okay, that's how they contain the structure, and they utilize the MADS domain for proper binding to the DNA, turning on a series of transcription events, making mRNAs. From the mRNAs, they will be making proteins, and the proteins and their interactions ultimately lead to the formation of different flower, not only flowering, but the MADs are there in many different aspects which are involved in flowering, the plant's growth, root growth, shoot growth, and many other aspects of plant development. Now, regulation of floral organ identity, particularly regulation of this gene expression in the floral organ identity. A key advance in the field of flower development is uncovering the direct role of another gene that is known as FM or floral meristem identity gene known as LEAFY, short form LFY. Floral meristem identity gene in the activation of the floral organ identity genes. Okay, so as I mentioned earlier that there are MADS domain proteins that act as a transcription factors. They go and interact to the DNA and initiate the transcription of specific region, translation, and the gene products are produced. But how they are regulated? Basically, the regulation is done by the floral meristem identity gene or LEAFY gene. LFI encodes a novel plant-specific DNA binding protein that can bind to the promoters of several floral organ identity genes. LFI alone is enough activate expression of the class A gene AP1 throughout the flower. Expression of each of the floral organ identity gene continues throughout most flower development. So LFI alone is enough to activate expression of the class A, that is AP1 particularly, throughout the flower. So if leafy is not working, AP1 will not be working. Okay? And when expression of each of these floral organ identity gene is done, then the response will be ultimately the flower development. The plant hormone gibberellin appears to be an important regulator of the homeotic gene expression at the later stage of the development. That's why we told you earlier the gibberellic acid is responsible for plant flowering, because gibberellic acid is a promoter for the leafy, particularly LFI, which is a part of the floral meristem identity gene, which in turn regulates the expression of floral organ identity genes. Okay, so gibberellic acid influences LFI, LFI influences floral organ identity genes, and floral organ identity gene products in turn are going to be participating in the process of class A, B, C activities, and along with that class E activities, which will regulate the not only the conversion of different roles into different specific type of structures, but also their position. Okay, that leads to the flowering in plants.